So, hello everyone, I'm Cesar Marin, and we are once again in the What We Are Reading YouTube channel of the South American Mycorrhizal Research Network. Um, Today we have with us Dr. Pehim Limbu. She's a postdoc fellow at Dartmouth University in the Balad Chaudhari's lab. And they recently published a really nice paper in PNAS entitled Climate Linked Biogeography of Mycorrhizal Fungal Spore Traits, with a really nice team of co-authors, including Sidney Sturmer and Carlos Aguilar Trigueros and some more. So, welcome, Smirti. It's very nice to have you here. Can you tell us what is the context of this study? Is this part of your postdoc? Can you explain us a little bit further on that?
Yes, absolutely. So first, thank you so much for having me. This is my first YouTube interview, and I'm really excited. Coming back to your question, so I am really interested about global change effects on plants and microbial communities. And my co-authors, Bala, Sydney, Carlos, and Jeff, like you mentioned, they also think about global change effect on fungal communities. So we had this great opportunity of looking at the fungal spore traits of arbuscular mycorrhizal fungi and to see if they show any climate-linked patterns. So this is how this study came about. And coming back to your second question, this is one of the central theme of my postdoc research, but I also do a lot of other work. Like I work with plant communities and plant microbe interactions in the context of global change. And I also study synchrony and coupling between these communities and as a result of climate variability. But this is one of the central research of my postdoc.
That's great. That's great, and it's also great that you are are doing more additional work. So I read the paper, and one of the first questions that you address in the paper is the importance of studying microbial traits. Can you elaborate on that?
Sure. So traits are these wonderful tools that we have, right? Like we can bridge the species community with their function and with their environment. And by studying traits, we can go beyond species and we can ask this question of like how will microbial communities respond to climate change and also management and different anthropogenic changes. And I don't know what your experience has been like, but my experience with microbial communities in the context of global change and restoration has been like microbial communities are this black box.
Mhm.
We don't really understand and we don't really know how to incorporate them in different global change studies and also restoration ecology. And I think like traits are fantastic tools because now we have the opportunity to understand mechanism and also test different ecological theories. So I think this is a great new avenue for advancing trait-based ecology and also global change ecology.
Yeah, I couldn't agree more. And trade ecology definitely has skyrocketed over the last 2 decades, I would say. There are plenty of databases for plants, for animals, for microorganisms, which indeed show this, as you say, this is a great tool to address these global scale questions. Can we go specifically and I probably will link Bala's paper on what are mycorrhizal traits in the description of the video, but can you go specifically on what are mycorrhizal traits?
Sure. So I think I would define mycorrhizal traits as measurable characteristics of plants or fungi or the symbiotic relationship between these two. For example, in the context of fungi, there are different traits like spore traits, right? And in the context of plants, there can be root morphology or photosynthetic pathway. And in symbiotic traits, there can be colonization rate that I often study as well. So I guess it depends on in what context you are trying to define the traits, but all these traits are wonderful, and they give us this framework to study these wonderful plant microbe interactions.
Among those you just mentioned spore traits, and this study was focused on spore traits. Um, so, which spore traits are important for arbuscular mycorrhizal traits, for arbuscular mycorrhizal fungi, sorry, and why? Why are those traits important?
So we studied five traits. We studied volume, ornamentation, cell wall thickness in relation to volume, shape, and color. And each of these traits, each of these spore traits has been found to affect ecological roles or expected to in AM fungi, right? Like there aren't many studies addressing that in AM fungi. And we found the same, like for example, volume had a role in germination, but also in dispersal potential, and similarly color, because they are more prevalent in warmer, wetter climates. Maybe they are protecting from UV, um, UV, and maybe also fire, right? So I think like these spores have ecological meaning to them.
Um, yeah, and this work was based in a database that Bala and others curated and published. I actually remember Bala presenting this in a conference that I organized in the Amazon in back in 2023, like presenting the database. So can you tell us how this collection of data is conducted, and what kind of sources, I mean articles, or I don't know, AMF collections compose the database?
Sure. So I think Bala would be the person to go to ask about trade AM, but I was a small part of it, and we leveraged all these taxonomic studies to look at different sport traits. For our paper, this PNS paper, we harmonized data across three major databases. The first is trade AM for sure. We also looked at global aim fungi database, and also Sydney has been collecting this wonderful biogeography database of AM fungi for a long time now. So we also leveraged that database and synthesized those together to ask these different questions.
And now you are going into the methodological part. I mean, is complex. Is a lot of work. So congratulations on that. Can you, yeah, very briefly explain the gist of the methods that you implemented? How you relate environmental variables with these five sport traits, sport traits, yeah.
Yeah, so I think the process started with these three large data sets, and we were like, what can we do? What are the questions that we can ask? And there was a lot of time spent in cleaning, harmonizing this database. Like we all know how time consuming that all is. But I'm very thankful for all the databases that I could use. And first of all, we standardized these data points across all the studies, and we also applied controls in our analysis to control for different methods, and also to control for different geographical potential geographical biases. And for environmental variables, because we had so many, and we wanted to um, give a clear message of how climate affects these traits. So we used principal component analysis to summarize those environmental variables and look at the different patterns of the environment before associating them with the traits. Hopefully that answered your question.
I think so. Yes, yes. This is very clear. And so you have these five sport traits, and then the classical environmental variables that one can think, like humidity or weather variables in general, or climate variables. Can you tell us, based on those two things, which were the main hypotheses that this study had?
So the first and most important hypothesis that we had was we will see the climate signature on these fungal traits. We also expected that these traits will predict species range sizes because traits influence dispersal, persistence, and survival as well. And we also expected that these traits would be affected by climate in terms of their trait diversity. So we expected that alpha diversity would be higher in resource-rich environments. And also we expected that if two sites are different in climate and environmental variables, then their traits would also be dissimilar to each other.
Okay. So what did you found regarding this hypothesis?
So, of course, there was clear climate-linked patterns in each of these fungal traits. For example, larger volume was more prevalent in warmer, wetter region than cooler, dry ones. We also saw that these traits were very good predictors of species range size. In fact, intermediate cell wall investment or the cell wall thickness was associated with the largest range size of the species.
Mhm.
So too thick or too thin, you are not able to maybe disperse more
Oh,
than the intermediate level of cell wall thickness.
oh.
And we also saw in terms of alpha diversity that richness was higher when they were in this environment of warmer temperature and higher precipitation.
That's so interesting. So, really, really small or really, really big spores have a smaller distribution, a smaller range, and intermediate sites have a broader distribution, right?
Mhm.
And do you know if these intermediate spores belong to certain uh, Glomeromycota genera or families, or they are everywhere?
Um, I think there some species have higher investment for sure, and that is the pattern that we are seeing. Exactly, I'm not sure which species. I'll have to look into the data and get back to you. But there
Mm.
are definitely some species that have higher distribution and also higher investment.
And there was another trait, a spore trait that affected the species range, or it was just the size?
Most of the five traits that we have did predict a range size. For example, volume had an inverse relationship with range sizes. So lower volume was associated with higher range size. And it makes sense, right? Like if you are larger, you are heavier, and maybe you cannot travel more.
One of the last questions that I have, and it's always interesting for me to know this, is between a single species, let's say Rhizoglomus irregularis, is there much variation of these traits, or they tend to be similar?
So, for the purpose of our study, we only looked at central tendencies, we only looked at mean. But when I was working with a trait aim database, that I know that there are a range of traits, right? Like some of the traits within a species vary even like two folds or even like three folds. So I
Mm,
think that is a very interesting next step in the study to
absolutely.
look at how intraspecific plasticity changes with environmental variability.
Absolutely. I remember seeing last year in a conference, a seminar by someone in Washington State showing that color, sport color. So she did a bunch of treatments, of fire treatments, different intensity and no fire, and definitely fire, of course, affected color, particularly sport color, quite significantly. So that's very interesting. I agree that this should be the next step. Um, yeah, you also in the results you have a section on phylogenetic diversity. Can you tell us a bit more about that?
So we didn't correct for evolutionary or phylogenetic history in our community weighted traits and their relationship with the environment, but we also wanted to see are there certain traits with higher conservatism than other traits. And what we found was indeed ornamentation was more phylogenetically conserved than other traits.
Oh.
And for example, investment was didn't show much phylogenetic signal. So there is a difference in these traits. Some are highly conserved and some are very flexible ecologically.
Oh, that's so interesting because that can also, I guess, inform future taxonomic efforts, knowing that some traits are very conserved in the phylogeny and some are not. So yeah, just to conclude, what's the main kind of conclusion or the main take-home message of this study?
So we saw clear signature of climate on these fungal spore traits. So I think what I would like people to take away from our conversation today is that traits are useful tools, and this helps us understand where fungi occur and how these ecological strategies might shift due to climate change and how that might affect our ecosystems.
Great. And personally, for you, what are your next steps?
I hope to contribute more to mycorrhizal science and also global change ecology for sure. I'm really interested to test these global trade climate relationships that we see here in experimental settings. So I'm really interested to test how different sport traits respond to different management practices, for example, grazing
Mm.
or fire, like you said from that study. And how we can use this approach in restoration ecology and different ecological systems.
Excellent.
So I'm planning to do that. Yeah.
Excellent. Thank you so much, Smriti. It was a pleasure talking to you and see you next time. Thank you so much.
And thank you so much, Cesar, for having me here today. And thank you so much for the YouTube channel as well. I go and look at your videos and I'm always inspired. So thank you so much.
Thank you.